me Professor Dr. Bansappa, Associate Professor, Department of Studies in Organic Chemistry, University of Mysore. The chair will be handling the session from now.
Good afternoon, everyone, to the session 7 on the second day. The first speaker for this session is Indrishesh Bhattacharya. He is from Department of Zoology, HND Dharwad University, Central University, and his topic for today is evaluation of season-specific differential gonadal architecture in adult Himalayan snow trout. And I welcome him to start the session.
Good afternoon. I am thankful to the organizer for giving me this opportunity for presenting my work. So I will be speaking on evaluation of season-specific differential gonadal architecture in adult Himalayan snow trout side effects. So other than the economic value, fishes are a very interesting model to study the gonadal development, right? Because they have both monopolistic species, one sex at a time, or hermaphrodite species, multiple sex at a time. You also have a high gonadal plasticity during the development. You have a cystic or synchronized mode of spermatogenesis in fishes, unlike mammals. And you have somatic supporting stem cells. You have certainly stem cells in fishes. You do not have it in mammals. And in fishes, you have two stem cells. And uniquely, you have fishes, they have a unique plasticity of germ-line stem cells. So if you go for germ-cell transplantation in the fish, the fate of the transplanted germ cell depends upon the recipient, not the donor. In mammals, it depends upon the donor, but in fishes, it depends upon the recipient. And also, there is a discovery for a new molecule in 5 years back, in FOXL3. This is the paralogous gene of Oxide, which is absent in the tetrapod. And if you knock it out, then it is specific to the female germline, and if you knock it out in adult ovary in the form. So these are the very critical things which makes fishes a very interesting model, other than the economic importance. So this is the crosstalk between HT and HTI axis. So basically, it is here important to know that these are the gonadotrophins, and the testosterone is actually bioconverted to either estradiol or into the active form of testosterone in fishes, that is 11 testosterone. So the bioconversion rate of testosterone to estradiol or testosterone 11 to testosterone, that will determine the sexual trait of fishes. And here these are the transcription factors. You can see FOXL2, DMRT1, these are the main transcription factors, which is actually changing the bioconversion of testosterone to estradiol in female and testosterone to 11 testosterone in male. So I have done some initial work with goldfish. It can be a very interesting model to study because unlike Medaka and zebrafish, if you deplete the germ cells in goldfish, they do not interfere with the sex. Generally, if you have more germ cells, the sex will be female in fishes. If you have less germ cells in gonad, the sex will be male. So this study actually shows that the goldfish can be a very interesting model for studying gonadal development. So we have done some microscopy analysis in adult goldfish gonad, and we have done this next-generation sequencing. And we have found out that there are various genes. Particularly, you have high DMRT1 has been expressed in males, whereas high FOXL2 is expressed in females. So these are some earlier studies that we have done with goldfish. Now coming to this current study, Cytothrax rhizostomus, which is known as Himalayan snow trout. So you can see the huge range of distribution from Iran to Myanmar, including the Tibetan plateau. But despite this huge range, it is a highly neglected Himalayan fauna. Only 187 heats you can get it in public. And also it supports the rural economy because it is a very important food source. But the phylogeny is actually the taxonomic classification is also distinct. So not that was what has been done. So aims and objective. First two aims and objective I am showing the data. Third is under process I am not showing the data. First one is to understand the phylogenetic status of cytoplasm. Second is evaluating the seasonal variation of gonadal architecture and function during the entire reproductive cycle. So first two things, the data I will be shown here. So here the first objective. So what we have done, we have collected these cytoplasm rhizostomas and we have sequenced the mitochondrial CO1 gene, and we have compared with different other CO1 deposited of different groups of cytoplasm deposited in the gene bank. So from this data you can see that this is the black box is our samples. So this is matching with the known reported cytoplasm rhizostomas, and they are much more closer to echinus, cytoplasm echinus. So these are the specimen of fishes which is showing, and it is much more genetically different than Cytoplasm Richardsoni. In Uttarakhand, in our river, Alaknanda river, Richardsoni and Cytoplasm rhizostomas, they are sharing the same habit. So I need to confirm that they are genetically distinct. So this data shows that rhizostomas is far more genetically different than Richardsoni, and Richardsoni is far more closer to Cytoplasm rhizostomas. Now here, these are all our samples, and we see that all cytoplasm rhizostomas, they show much more genetic proximity when you are taking up one single river and stream. Like all fishes, rhizostomas in Alaknanda river, they are much more closer. And you have differences when you have different streams like Bhagirathi, Nandaki, Yamuna, Jhelum, Express. And here we have taken the Cytoplasm rhizostomas with all other genera under this family Cytoplasm. So our sample Cytoplasm rhizostomas come in this particular group, which is actually noted by the primitive Cytoplasm group. So here this phylogenetic analysis actually establishing that what is the phylogenetic status of our model organism Cytoplasm rhizostomas using mitochondrial CO1 as a molecule. Next is the AM2, what is the seasonal reproductive cyclicity. So you have the immature and then you have mature, and this is the reproductive cyclicity, degenerative stage, developing stage, spawning capable stage, and regressing stage. So this is the experimental design. So we have freshly captured Cytoplasm rhizostomas. Then we have used DNA barcoding to identify the species identification or physical identification. Then we have measured the GSI, and also we have quantitatively analysis. In case of male, we have done the spermatic sperm density. Female, we have done the fecundity analysis and the ultrastructure by light microscopy, transitional microscopy, and scanning microscopy. So these are the dissected gonads. These are the GSI. So you have two reproductive peaks here, February, March, and September, October, and you can see there are sexual dimorphism here, facial tubercles in spawning males. So in these are the testes developing, spawning, and regressing. So here you can see the most of the cases you have differentiated or undifferentiated spermatogonia or primary spermatocytes. You have beautiful sperms and spermatids here, and you have some residual sperms in the regressing stage. And these are the structures of these sperms, and clearly see we do not have the acrosome and very beautiful images. Similarly, in ovaries, you also have two peaks, February, March, and September, October, the GSI values, and again you can see there is a change in the developing spawning capital and regressing phase. The number of eggs are different. Now, in case of developing, you mainly see the immature follicles. You can clearly see the germinal crystals here. In spawning capable phase, you see this microgenic oocytes, and also you can see the cortical alveoli. And regressing phase, mainly you see the post-ovulatory follicles, and also you can see some acidic follicles. Same thing here, you have mature oocytes, you have mature microgenic oocytes, and you have the acidic follicles. So, having said that, what is the conclusion and take home? This RNA-seq data in goldfish and histological data in cytoplasm stages establishes them as a suitable model system. Future studies will employ the de novo transcriptional sequencing in sex and season-specific manner to investigate the regulatory genetic network critical for productive chemical in cytoplasm. I am thankful to Dr. Mahendra Singh for collaborating in this DNA barcoding analysis. The Reproductive genomics for maturation sequencing aims for microscopy and my students share Pratibha and Sachin. And funding from UGC and DSC IE3 level. And last, I'm thankful to HRPC organizers and special thanks to Professor Chandranagan Madam and Dr. Suresh sir for kindly nominating me for this prestigious Timari Award given by the society. Thank you very much. Thank you. Good afternoon. I am thankful to the organizer for giving.
Okay, the session is open to questions.
Hello, can you hear me?
Yes, I can.
What is the rationale of using CO1 or cytochrome B for phylogenetic relationships? Why not nuclear gene or standard rRNA genes?
No, actually, CO1 and cytochrome B is more popular. Thereby, we are using it. And recently, they are using RAG1 and RAG2. So we are continuing this work, both nuclear as well as the mitochondrial gene. So we have done with 2 markers so far because it is far more well studied. So from gene bank, we are taking the deposited samples, which is available for CO1 and cytochrome B. RAG1 and RAG2, we are just initiating.
Okay.
Any more questions? So if no questions, so we move on to the next speaker. So I thank you, Indulgence Bhattacharya ji, for giving a very good talk in this online class.
Sir, can you announce to Raro Mr. He is unmute.
So in this session, second lecture, online lecture. So I welcome Dr. Raghav Kumar Mishra. So who is an associate professor in Department of Zoology, Banaras Hindu University. So we welcome you, sir. So the title of the talk, today's talk is Effect of Psychological Stress on Male Potency. So sir, please take the session.
Okay, thank you. I'm thankful to organizers for inviting me for this. I would also like to thank my teachers who are present online, Professor Chandra Algar ma'am and my PhD thesis examiner, Professor P.P. Mathur sir. So I'm going to present my talk on effect of psychological stress on male potency. So as we know, reproductive health is fundamental to the society and economic development of the community. Not only for that, good reproductive health is not merely absence of a disease. It is giving a feeling of satisfaction for the emotional health, well-being, physical, psychological, and mental health to not only to the individuals but to the couples and to the families also. So nowadays there is an increase in male reproductive disorders, male sexual disorders, which are broadly classified into two categories, that is the infertility and impotency. There are many causes for infertility. The major causes are low sperm count and sperm abnormality, which is directly related with the spermatogenesis. So it has been observed in the last few decades that out of total number of couples who are failed to parent a child, 50% cases in those couples is due to the male factor infertility. And this male factor infertility is having a negative impact on a relationship, particularly the marital relationship, and it causes conflict, and sometimes in the worst condition, it goes towards the divorce or divorce also. The second most important nowadays, which is major disorder of the male reproductive male sexual disorders, is impotency. So, which is marked by the low sexual desire or the erectile dysfunction. So, it has been observed that there is a huge increase. Number is increasing day by day, because I was reading the WHO report where it was in 90s, it was 150 million. Now they are expecting that more than around 325 million people, men, are affected with the erectile dysfunction. And it is also suggesting that nearly 15% of males, they are every year they are suffering from this male sexual disorder. We can define male erectile dysfunction as a inability to achieve and maintain the erection for sexual intercourse. Earlier, we used to talk about the Indian subcontinent, but it is not the Indian subcontinent. Everywhere in the world, erectile dysfunction is having a marked impact on the individual's social, psychological, family, and all aspects of his life. Because if society comes to know that a person is suffering, a person is impotent, then the society is started treating that man as an incomplete man. Pardon?
Your slides are not rolling up.
Slides are not rolling.
Yeah, can you please share the slides so that we will do it? Down there. Yeah, fine.
Make that one for slides.
Slide mode.
Yeah. Slideshow mode.
Notice
Presentation mode. That is in down.
Right corner of you. So
Full screen mode. Make it full screen and move the slide along with your talk.
presentation mode क्यों नहीं जा रही है?
The slides are not in full screen mode.
ನೀವು
You
ತೆಗೆದು
will press F5, sir.
ಶೇರ್ ಮಾಡಿಲ್ಲ ನಾಟ್
This way it is moving.
No, it's moving, but we can't see it full screen. Please press F5. Down arrow.
ಯಾ
Now it is moving.
It's moving, sir, but we are seeing it in the PowerPoint mode. We want to see it in slideshow mode. Your slides are seen along with the full slide list.
This way you want.
Slideshow.
क्या हो रहा है?
It's okay, it's okay.
You need to go for the supply structure is there. Or otherwise make it 100%.
So just beside the 100%, there's a small tab. If you can
It is on.
shift F5 will also work. So we can continue.
Now it is moving.
So it's okay, we may continue.
Okay, so should I continue?
Yes, sir.
Okay. So I was talking that impact of erectile dysfunction on society, because society started treating the person who is suffering from erectile dysfunction or impotency, they started treating as an incomplete man. Now, so what are the main causes nowadays in the erectile dysfunction? The first one is the low testosterone level. As we know, that testosterone is crucial for the libido. So low testosterone level is interfering with the nitric oxide release, and that's why it's causing the erectile dysfunction. Another is smoking and alcoholism. As we know that both are affecting the hypothalamic nuclei, and then that's why they are down. They are affecting the down the pathway of this penile erection. Now, another one is age, which is very crucial. As we know, with the age, there is decrease in the testosterone level, as well as in other factors which are regulating the penile erection. Now, blood pressure, as we know that blood pressure, high blood pressure patients, which are generally having high blood pressure, they may develop a problem which is called as atherosclerosis, and that may cause erectile dysfunction. Another is the lifestyle. Nowadays, as we are seeing that there are a lot of alteration due to job or other factors, there are a lot of alterations in the lifestyle, and which causes the erectile dysfunction. But most important is that the psychological factors which are associated with the erectile dysfunction or physiology of penile erection that is causing. And what are these factors? They may be personal ambitions or workplace demand and social or family support, frustrations due to the work, or the other factors, relation challenges. Because of these factors, it is now considered that psychological stress is now considered as a byword of today's life. And before COVID, it is not present data. Before COVID, it was 21%. It was observed that 21% of men are suffering from psychological or under the psychological stress due to one or the other factors. And this present pandemic COVID-19 is also adding to this psychological distress. So this psychological stress is known to hamper the male reproductive system, and they are causing male infertility by acting on this, by disturbing the spermatogenesis through oxidative stress mechanism in the testes. But what is the impact of this psychological stress on penile erection is not fully known. So our aim was to understand how psychological stress affect the penile erection, and for that we are using restraint stress process method, in which we are immobilizing the rat in a restrainer, and this restrainer is giving because of this immobilization, they are under psychological stress, and they because of physical discomfort and others, they are under the psychological stress, and that is established model which can mimic with the psychological stress of human beings. Now, before going for effect of this restraint stress on male potency, first we should know that what is the pathway of regulation of male penile erection or male potency. The first is that if after receiving the stimulus, the dopamine is released, which is binding to the oxytocinergic neurons bias due to receptor, and then by the interaction of calcium and clavulin, NOS is synthesized in the nerve endings, and these are then catalyzes the nitric oxide synthesis. This nitric oxide enters into the smooth muscles of the penile tissue, where they activate the guanylyl cyclase, and which activate the GTP and convert it into the cyclic GMP. And this cyclic GMP activates the PKG, or protein kinase G, which is altering the calcium channel and potassium channel, causes relaxation in the smooth muscle cells. Meanwhile, what happens? Because of this relaxation in the smooth muscle cells, there is an erection. So that's relaxation invites the blood flow into the vein, arteries of penile tissue. So this force of blood flow activates the PI3K and AKT proteins, which phosphorylate the endothelial nitric oxide synthesis, which are synthesized in the endothelium cell because of acetylcholine, which binds to the its receptor in endothelial cells. So these eNOS will give rise to nitric oxides. So both nitric oxides from eNOS and nNOS, they are responsible for this penile erection, and mainly it was triggered by the nNOS, and then it is maintained by the eNOS. Now, to study the impact of psychological stress in animal model, we have used an restraint stress model, and we used SD rat, and we placed the rats into these trainers, and for different durations. And we have collected, based on our previous experiment, we have collected 1.5 hour and 3 hour durations for exposure to this restrainer stress, and we have evaluated this following parameters. Now, after exposure to restraint stress, as we observe that there is a sharp decline in the sperm count. However, if we talk about the motility and viability, there is a decrease, but that decrease is not significant. But the sperm count is significantly decreased in case of restraint stress exposed rats. Same is true for the sperm morphology also. Different types of morphological structures. We have abnormal structures we have observed, and most common is that head abnormality and tail abnormality. And as we observe that the percentage of abnormal spermatozoa, the tail abnormality are more than the head abnormality, we observe even the two head spermatozoa in our restraint stress exposed rats. Now, this exposure to restraint stress also decreases the testicular daily sperm production, which is an indicator that this stress is affecting the spermatogenesis in testes. Now, this also have negative impact on testosterone. Also, although it is not very much significant, but there is a dose duration dependent decrease in the testosterone level in restraint stress exposed rats. Now, we want to know the impact of this restraint stress on potency. So we observe the sexual behavior, and we observe prosexual and sexual both behaviors, and we observe that there is a decrease in non-contact erection because we perform a non-contact erection test to the experimental animals, where we place a transparent plate with a lot of holes, so that there will be auditory, visual, and hormonal signals will be there, but there is no contact. So in that case, we observe that there is a decrease in non-contact penile erection in the rats which are exposed to 3 hours for restraint stress. When we observe the sexual behavior of these rats, then we observe that there is a decrease in mouth, but that is not much decrease in the mouth, but there is a decrease in intromission, ejaculation, and intromission. We observe that there is a sharp decrease in the ejaculation ratio of rats which are exposed to the restraint stress, which is a type of psychological stress, and there is also a decline in the intromission ratio in rats exposed to the longer duration, 3 hours of restraint stress. we check the neuromodulators because as we know that neuromodulators, penile erection is regulated by the neuromodulators. So we check the concentration of these neuromodulators in the serum, and we observe that there is a slight increase in dopamine in 1.5 hours and oxytocin in 1.5 hours exposed rat, but there is a decrease in serotonin in 1.5 hours exposed rat.
Can you conclude, please?
Yes, please.
Please, can you conclude? Your time is over.
Okay, just okay, concluding. So same is true with the when we observe the eNOS expression in the penile tissue, we observe that there is a sharp decline. In this case, you can see the 3 hours because this is the control. So if you see the 3 hour, there is a sharp decline in eNOS expression in the epithelial cells of the penile tissue, and then there is also a sharp decrease in the expression of nNOS in the epithelial tissue in stress exposed rats. We also check the nitric oxide level as we observe that there is a decrease in nitric oxide, which is a regulator of penile erection, and there is a increase in PD5A, which is acting, which is responsible for deactivating the cyclic GMP, and there is a decrease in PAKT level. Now, in conclusion, what we observe that the restraint stress is decreasing the testosterone concentration, which adversely affect the sexual behavior, prosexual and sexual behavior, the sperm parameters and testicular daily sperm productions, which is responsible for sexual dysfunction. Meanwhile, it is also affecting the neuromodulators in the hypothalamus, which affect the sexual behavior, and as a result, there is a decrease in PAKT, nitric oxide, and there is increase in PD5A level, and this affects the sexual dysfunction, which results into the impotency. So we can say that the restraint stress exposure decreases the expression of eNOS and nNOS, PAKT, and nitric oxide level in the penile tissue, and increase in PD5A, suggesting that this psychological stress is NO and PD5A dependent separation of this are causing impotency in mammals. Thank you.
Thank you. So, on behalf of the organizing committee and on behalf of the conference, we thank Mr. Raji for his good talk. So now the session is open for discussion. Any more questions? One or two?
Rakesh, how do you establish the psychological stress? Have you used any criteria or any inclusion and exclusion criteria you followed in the rat or the human? Wherever you establish the psychological stress, can you highlight it?
Actually, sir, we have used G-strain stress model for developing the psychological stress, which is established model for mimicking with the psychological stress of human beings. And we can extrapolate our data from animal to human. That is the established model. So we observe that because we observe here that in case of rat, when we put in the restrainer, we are actually immobilizing them. So by immobilizing them, we are giving them a type of stress which is equivalent to psychological stress in the human beings.
Thank you, Raghav. So we'll move on to the oral presentation by Seema Rai. So
I hope I am audible to all.
yes, so
Sorry.
Seema Rai is from Department of Zoology, Guru Ghasidas Vishwavidyalaya, Central University. So today the talk of Seema Rai is entitled Combined Administration of Melatonin and Insulin, which improves diabetic-induced impairment of rat kidney function. So, these are.
Respected all the dignitaries on the dais of the dais. Very good afternoon to one and all. Today I am going to share my some of the research finding under the topic combined administration of melatonin and insulin, which improves diabetic-induced impairment of rat kidney function.
Dr. Rai, can you put your screen on slideshow mode?
Is it okay?
Yes.
So as we all know that diabetes is a complex metabolic disorder, as well as it is epidemic for all the public health concern. The increasing rate of diabetes in modern society include the change in the lifestyle, working pattern, as well as our food habit. Recently, the International Diabetes Federation, they have reported that diabetes is responsible for the death of more than 6.7 million. That is reported in 2021. That means that one death in every 5 diabetic patient. So many speakers have been talked already about the melatonin, their function, and we all till date know that it's a pleiotropic effective molecule which regulates many of our functions, including sleep-wake cycle, reproduction, biological clock, rhythms, central peripheral synchronizer, or as well as it is the nephroprotective. It is a very good free radical scavenging molecule, as well as recent finding also suggests that due to low level inflammatory status, diabetic patient, recently it has been observed that they are more susceptible towards the SARS-CoV infection. And this melatonin can also be used for the treatment of those patients. Coming to the second molecule, that is the insulin, which is secreted by the beta cell of the endocrine pancreas and then regulates the blood glucose metabolism in different organs, especially the liver, the adipose tissue, and the muscles. So it is well established that diabetes-related metabolic disorder due to hyperglycemic conditions, they lead many of the renal functions. And there are very allopathic drugs which are available in the market for the treatment of the diabetes, but they somehow produces many side effects. And they are not recommended for the cure of this disease. So looking into all these things and their side effects, this hypothesis has been proposed in search of the two combined molecules, that is the insulin as the previous treatment, as the melatonin as adjuvant treatment, to correlate, to establish where the combined effect of these two molecules can be able to restore the and recover the biochemical, hormonal, and other function to maintain the glucose homeostasis in any diabetic model. For this, we have used only the kidney cortex because the 85% of the nephrons are localized in the cortical region of the kidney, and synthesis of glucocorticoid also occurs in the kidney cortex. The streptozotocin has been used for the induction of diabetic model. This streptozotocin is a very is a diabetogenic drug which is synthesized by a bacteria Streptomyces acromycins, and this drug actually resemble with the glucose. With that, it actually hence it is mimicking the glucose and interferes with the GLUT2 receptors and leading to the diabetic conditions. That is the mechanism pathway by which we have opted to induce the diabetes condition. This is the total protocol of our study. We have the 54 animals, out of which 24 we have separated in a one group and 30 in one group, to 30 animals, we have injected 50 milligram per kg body weight, the streptozotocin for 6 days, and we observed continuously for their hyperglycemic condition for the complete successful induction of the diabetic condition. And then these all groups were divided into randomly divided into following these groups: controlled diabetic treated with the melatonin, then insulin, diabetic rats treated with melatonin and insulin. Then one treatment we have also compared with the glibenclamide, which is the clinically recommended anti-diabetic molecule. Another is with melatonin to see the per se action as well as with the insulin. So this is the successful induction of the diabetes condition we have observed up to the 28 days of whether there is any reversal or not. And we found no reversal was there after giving the 6 days of this streptozotocin. And the minimal level of glucose, blood glucose, we have observed 250, which were the standard for considering the rats as the diabetic. The further confirmation we have observed with the destruction of the beta cells and other cells in the endocrine pancreas. This paper we have also published. This finding also we have published in 2018. Then the parameter we have studied were gravimetric, biochemical analysis, histology, and receptor assay. These are the protocol, a standard protocol we have followed with some modifications. The results, first of all, we show that the maximum restoration we were found after the treatment of the melatonin and insulin in the restoration of the blood glucose level. Then body weight, we have not seen a very high significant changes. Only very low significant change was noted in the diabetic condition. Next was the renal function test. In renal function test also, diabetic rat displayed the considerable increment in the biochemical indices, and there were augmented creatinine, uric, and urea. Melatonin and insulin administration, they partly counteracted alone, but co-administration were appeared the more protective towards the diabetic induced renal cortical injuries. This is the histological histoarchitecture of the kidney, and we can see the degeneration during the diabetic condition. There were more, this is the diabetic condition, and we can see the restoration of the cortical granules and glomerular damages during the combined treatment of melatonin and insulin. And this is their tabulated information regarding the degeneration of the tubules, rate of glomerular damages, coagulation, and glomerular space status. Again, we have also looked for the anti.
Request you to conclude.
Okay, we have seen the similar kind of effect in the antioxidative enzymatic status, for which the co-administration appears to be very potent. Glycogen status also showed noteworthy increment after the co-administration of the melatonin, and GR cycle also have the similar kind of, when the co-administration have been given, they showed the integrum recovery in the GSH dependent antioxidative enzymatic activity. The serum cytokines are the more interesting. The diabetic rat showed elevated inflammatory cytokines like TNF alpha, IL-6, IL-1B, TGF beta, and however the IL-10 were found decreased. Then, because this is the anti-inflammatory and indicating an altered innate immunity and inflammation during because of which may be associated with the insulin resistance. This reason also may be because of the low circulatory level of the melatonin as well as the insulin during the diabetic condition. These are the relative expression of the MT1 and MT2. So we can see that during the diabetic condition, there are upregulation of the MT1 and MT2. But however, when there have been the combined treatment has been given, there are more control towards the expression. So this we can see that overexpression during the insulin and glibenclamide could be correlated with the low circulating level of the melatonin. So these are the major findings because of the antioxidative enzymatic status or free radical scavenging capacity of the melatonin. These things might have happened when there is finally, I would like to conclude that the favorably safe nature and nephroprotective property of melatonin and insulin suggest it to be a pharmacological adjunct to increase the therapeutic window of important nephrotoxic drugs in clinical trials. Thank you. I am thankful to everyone, especially to the organizer and my mentor, Professor Chandana Haldar, who I can see in this screen also. Thank you all.
Thank you. I think we can have one or maximum 2 questions.
Hello, is my voice audible?
Yes, sir.
Yeah, thank you, Dr. Seema, for a good presentation. I want to know that the nephroprotective role of melatonin is well established. So, in your presentation, like, what is the take-home message? What is new in this?
Sir, I have correlated the things with the rationale with the expression of the melatonin. Actually, many of the part I was not able to include because of the shortening of the time. Many other things we have also done related to the expressions. So that is the more important part.
For the nephroprotective role of melatonin,
it is associated with the regulation of MT1 and MT2 receptors, sir.
So which part of the kidney you need to say it's like? Is it the because see protection you have reduced the damage to the nephrons, so it is the convoluted tubules are damaged. So how do you think the melatonin is imparting protection to that?
So because of its antioxidative action, as well as it is also enhancing the antioxidative enzymes to the body to combat with the free radical which are produced during the hyperglycemic conditions.
All right. Thank you.
So, madam, what is the concentration of melatonin at that time?
Sir, pardon, I am not able to hear.
What is the concentration of melatonin when you are treating over here?
It was 200 microgram.
200 microgram.
Yes.
Hello.
Yes, sir.
You have given 15 milligram per kg body weight Stressor, am I right? To induce diabetes. 15 milligram per kg body weight. Am I right?
Sir, sorry.
Hello.
Yes, sir.
Is it audible?
Yes, sir.
You have given 15 milligram per kg body weight Stressor to induce
Sir, streptozotocin has been given 15 milligram per kg body weight.
Yes, that's what I am saying. It is quite insufficient to induce destruction of beta cells, right? That too, the duration of 6 days is not enough to have a sustained diabetic condition. And number 2, you said you have fixed a criteria of 250 milligram per deciliter of glucose to consider it as a diabetic condition. But your graph shows very much less than that.
No, sir. It is actually the graph and as well as my table. Actually, there were tables which were showing higher than 250. This is the minimal concentration which I said that we have considered this concentration to to keep that if any animal has 250 and more than 250, those were considered as the diabetes animal. But in our case, we noted more than 300.
More than 300.
Yes, sir. It was in the table of my presentation.
But I have my own doubt because 15 milligram per kg body weight is not okay. Please go through the literature. Right? If you come across any paper, it says that 15 milligram per kg body weight is quite sufficient to destroy beta cells and can induce diabetic condition with 300 milligram per deciliter glucose. I will be happy. Please check. Okay.
Okay, sir.
Okay, thank you, Dr. Simalan. We now move to the next oral presentation. One instruction for all oral presenters. We stick to 8 minutes per presentation, 2 minutes for discussion. The next presenter is Jiva S.E. from Molecular Reproduction Unit, Rajiv Gandhi Center for Biotechnology. And his topic is evaluation of the role of BYNLT1B in stemness, spermatogenesis, and infertility.
Good afternoon, everyone. I'm Jiva SC from Molecular Reproduction Unit, Rajiv Gandhi Center for Biotechnology. I'm here to present part of my PhD work titled Evaluation of the Role of DYNLTB in Stemness, Spermatogenesis, and Infertility. Let me start with the brief introduction of spermatogenesis. As we all know very well about the spermatogenesis. Spermatogenesis is a complex process takes place in the seminiferous tubules of the testis, where the spermatogonial stem cells undergo self-renewal and differentiation.
Can I request you to put your slide on the slideshow mode?
Actually, it is already.
Yeah.
It is already in slideshow mode.
No, we are not able to see it.
Presentation mode.
Shift F5.
For me, it is showing in 1 minute. Is it okay now?
No, we are still seeing a. Can you share the whole screen? Okay, it's okay. Continue. Please continue.
So the spermatogonial stem cell in the testis undergo self-renewal and differentiation through a series of mitotic division, and further it differentiate into mature spermatozoa through a series of meiotic division and morphological transformation. Any defects in this molecular regulation will result in loss of germ cell or will result in impaired spermatogenesis, which is considered as one of the important factors for male infertility. Coming to the background study, our lab mainly works with the genetic factor causes of male infertility. The previous study conducted in a lab between the fertile and infertile subjects identified a lot of candidate genes, and dynein light chain 1 or DYNLT1 is one among the genes. We further evaluated the expression of dynein light chain 1 in human male germ cells. From the image, it is clear that both at the message level and at the protein level, there is an aberrant expression of dynein light chain 1. And we associated this aberrant expression of dynein light chain 1 with human male factor infertility. So we went to check on what is known about this molecule dynein light chain 1, which is also known as TCTX1. It is the gene this gene code for a 14 kDa protein named dynein light chain protein. It is a component of the dynein cytoplasmic dynein motor complex, which involves in the transport of cellular cargo. It involves in linking the cargo to the dynein motor complex and other proteins which regulates the dynein function as dynein dependent function, and also it involves in dynein independent function in neural morphogenesis, kinetochore assembly, and mitotic spindle orientation, etc. The human homologue, the mouse homologue of this gene is located in the chromosome 17, which is considered as a T complex region. Any genes in the T complex region is known to have essential for normal spermatogenesis in mice. Any defect in this T complex genes were reported to have spermatogenic impairment. Also, the expression of the gene is very high or predominant in the germ cells than other cell types in the mouse. Other reports about the gene includes its expression is enriched in the stem-like cells or in the cycling progenitors in the mouse brain in both adult and young and adult mouse brain. The chromosomal location and the germ cell predominant expression make this gene an ideal candidate to for the infertility study. So this prompted us to ask the question that whether this gene dynein light chain 1 is necessary for the maintenance of germ cells in the adult testis or not. To address the question, we have this following aim and objective, which is to understand this role of this gene in germ cell self-renewal and differentiation.
Can you change the slides? We are not able to see it.
Actually, for me, it is working perfectly. I'm not sure. I can change the slides. Is it changing?
I think, can you stop the share and share again?
Yeah, okay, fine.
Without much waste of time.
Can you see now?
Yes.
But for me, it is not working.
It's okay. Please continue. We can see the slides changing at least.
Yeah, fine.
Please continue this way. It's okay.
Okay, so the background literature asked us to ask the hypothesis that this gene dynein light chain 1 is necessary for the germ cell maintenance in the adult testis. To address the question, we have this following objective, aim and objective, which includes the understanding the role of DNLT in germ cell self-renewal and differentiation, and to evaluate the changes in stemness and germ cell marker with and global proteomic change with respect to DNLT1 manipulation. We evaluated the homology between the human and the mouse dynein light chain 1. Both shows 95% sequence homology at the amino protein level. So we further went to evaluate the expression of DNLT1 in different stages of germ cell development. From the image, you can see both RT-PCR and that Western blot. When the germ cell matures or when the age of the age increases, the expression of the dynein light chain increases with both RNA level and at the protein level. We further went to evaluate which population of the testis express this gene, and from the IHC image from the day 16 to adult testis, it is clear that the differentiating population in the testis expresses this gene predominantly when comparing to the undifferentiated cells and the completely differentiated cells. This suggests that the cells which are expressing could be the differentiating population or the differentiating progenitors of the testis. We further went to evaluate the molecular function of this gene by both gain of function and loss of function studies. For the gain of function study, we cloned this gene into the EGFP-N1 expression vector, and the clones were further confirmed by sequencing, and the clones were used for overexpression studies. The dynein light chain 1 overexpression construct was transfected into the GC1 germ cells, and then the overexpression was confirmed by both confocal microscopic image and studies and Western blot also. Here you can see the fused protein, dynein light chain and GFP fused protein from the overexpression construct transfected clones. And then this is the wild type GFP. And then this is the endogenous dynein light chain 1. And then beta-actin is used as the loading control. We further evaluated the expression of various stemness and germ cell factor with respect to dynein light chain overexpression. The image panel has 3 biological replicates of untransfected GC1 control, dynein light chain overexpression, and then empty vector transfected control. You can can be seen that the sum of the pluripotency factors like NANOX, SOX2, CMYC, LIN28 were upregulated with respect to overexpression, but KLF4 were downregulated. Similarly, the some of the germ cell factors like GFR alpha, DPPA3, GBX1, TEX19 were upregulated, and some other factors were downregulated with overexpression. We further quantified the upregulation and the downregulated. From the graph, it can be seen that with the overexpression of nearly tenfold overexpression of dynein light chain, the respective upregulation of the stemness and germ cell factors with respect to overexpression. We further went on to study the knockout of this effect of the knockout of the gene in the GC1 germ cell. We used commercially available CRISPR-Cas9 knockout vectors to knock out this gene, and we confirmed the knockout in both RNA level and at the Western blot. You can see the the truncated RNA, and also the complete loss of dinyl light chain 1 expression at the protein level. So we further evaluated the expression of stemness marker with respect to the dinyl light chain knockout. The markers which were upregulated with dinyl light chain overexpression were downregulated with dinyl light chain knockout. Here the nano is downregulated, SOX2 is downregulated, but CMYC, which is downregulated with overexpression, now upregulated with CMYC was upregulated in both overexpression and knockout, but KLF4, which was downregulated with overexpression, now it upregulated with knockout. So this is the quantitative graph represents the quantitative data of the image. And then here we can compare, it is compared both the overexpression and the knockout. So when dinyl light chain is overexpressed and dinyl light chain knocked out, you can see the expression of these molecules. Yes, ma'am?
Yeah, time is up, so you can conclude now.
Okay. Then we went to evaluate the global proteomic change with the overexpression. The overexpression proteome identified 114 protein are upregulated, and among the upregulated protein, the major class of upregulated protein include chaperone, cytoskeletal protein, and nucleic acid binding protein. We identified some of the three interacting cluster among the upregulated proteins, and the majority of the proteins includes the upregulated protein belongs to the Wnt signaling pathway, and then the cytoskeleton regulation by Rho GTPs. Both these pathway are essential for the differentiation of progenitors. Then some of the genes upregulated with overexpression were associated with spermatogenesis and mitotic cell cycle. Coming to the summary, the dinyl light chain expression increases with the germ cell maturation, and the expression is dominant in the differentiating population, which could be the progenitor population. The pluripotency markers like NANOX, OX2, CMYC, LN28 are upregulated, and also the germ cell markers like DPPA3, TEX19, GVX1 are upregulated. The same way, those who are markers which are upregulated with overexpression were downregulated with the dinyl light chain knockout, except KLF4. The overexpression identified 125 and differentially proteins, which are mainly belongs to the class of chaperones and Hitchcock proteins, cytoskeletal and reversible protein, which were associated. Many of these proteins were associated and known to involve in the spermatogenesis process and mitotic cell cycle regulation. My ongoing activities includes the germ cell marker evaluation and global proteomic change with dinyl light chain knockout, and future studies includes the transplantation study to evaluate the differentiation potential and fertilization capacity of the dinyl light chain manipulated GC1 germ cells. These are my publications and acknowledgement, and thank you.
Thank you. Session is now open to a couple of questions. Thank you. If there are no questions, we can move to the next presenter. Again, a request to all the presenters, please share your entire screen so that we can see the slides in full screen mode.
Next speaker, so I welcome Irfan Khan. So he's from affiliated to Molecular Reproduction Unit again, Rajiv Gandhi Center for Biotechnology. So I welcome you. So today your talk entitled with CNNM1 regulates spermatogonial stem cell maintenance and phase determination. So please start your presentation.
Good afternoon, everyone. So I'm myself Irfan Khan, working under the guidance of Dr. Pradeep Kumar G from the Molecular Reproduction Unit of Rajiv.
Please unmute yourself. You're not able to hear.
Hello.
Yes.
Okay. So reproduction is a fundamental and essential process where a male produces a functional sperm and a female produces a ovum. So these both fuse together to generate an offspring. So any defect in this process might lead to the infertility. So And infertility is one of the global alarming concern, which is though the people 15% of couple who are approaching for the ART treatment are not successful, because though causes of when we talk about the causes of infertility, that female factor contributes to the 50% and male factor contributes to 50% of infertility. Though the normal semen parameters are normal, the 30% of cases are still infertile due to lack of proper studies where the subcellular defects are not yet really clearly understood. So we need to have a disciplinary approach to really define what fertility is. So we need to have a proper proteome mapping of a sperm proteome, so that we could understand the subcellular defects that are happening before the production of a sperm. So our lab has performed the differential display proteomics and identified this subset of factors, that subset of protein markers, which could be appropriate way to define what defines the fertility is, and to devise successful therapeutics for the treatment of infertility. So CNNM1, which is also called a cyclin M1, is a protein of my interest, and it is one of the apparently expressed protein in the found it in the differential display proteomics, and it belongs to a family of protein called as ACDP with the 2 CBS domains, which is also called as CVS domain. So before we are using do a functional evaluation of CNNM1. So we are using a Swiss albino mice as a model. So here we are before going into the functional evaluation, we have to know how chronology of spermatogenesis happens in a mouse. So the 6.5, the PGC specification happens, and the gonocytes are formed at the 12.5, where they are during the programming phase. The SSC fate happens, where the SSC population is formed in the establishment phase, whereas the other subset of population will upregulate the differentiating genes, and which follow into the first wave of spermatogenesis. So with this, we did multi expression study of this mouse cyclin M1 in multiple tissues, and we found out a strong expression of CNNM1 in brain and testes. So as we are more interested in the reproductive part of a CNNM1, we further went to profile the CNNM1 expression in the postnatal stages during the development stages from 1 to 12 weeks, and where we found out that expression of CNNM was more in the first week. Then we went to a localization study, where we found out that at the 8 days it is more scattered expression, while at 16 and 24 days mice showed a very organized expression of cyclin M1 towards the basal lamin. So basal membrane. So usually the progenitor cells are more located towards the basal membrane. So we thought like maybe we further went to check its expression during the differentiation path dependent expression. So we did a co-localization assay, where we used a c-kit and OCT4 OCT3 to co-localize with the CNNM1, and we found that the expression is more strong towards the spermatogonial stem cells, which are OCT3 positive. To further evaluate this, we did this. We isolated the spermatogonial stem cells through the differential plating method, and we found that cyclin M1 expression are more in the spermatogonial stem cells, and when we retrieve the LIF and GDNF they formed the embryoid body, where the CNNM1 expression reduced. So as we saw that its expression is more in the SSC population, so we further went to check its expression during the PGC2 germ cell specification, and we found out that the expression increases after gonad formation, that is after the 12.5 after the post day 1, we found the increase in the CNNM1. So further to evaluate how this expression is in the gonocyte to the spermatogonia specification, we did an expression study, where we found out that the 3 days of population where the self-renewal of this SSC founder cells happens, the CNNM1 expression increased. So as we saw that the self-renewal during the stage of self-renewal, the CNNM1 expression increased. So we used it as self-renewal factor and differentiating factors like we found out that there's a self-renewing factor, the GDNF, increased the CNNM expression, while the treatment with a differentiating factor, RA, decreased the CNNM1 expression. As self-renewal is about proliferation, we used a letrozole, which is a male germ cell proliferation agent. So we found out that upon a letrozole treatment, CNNM1 increased the expression, while ginsenoside RH2, which is an anti-proliferative agent, decreased the CNNM1 expression. So this provides a clue that CNNM might be having a role in a proliferation and self-renewal. So further, we knocked down a CNNM1. So we found out they altered the cell cycle by decreasing the G1 population. So further, to evaluate its functional, we overexpressed the CNNM1 and we did a global proteome mapping of this. So we found out these are the biological processes that are upregulated with the CNNM1 overexpression, and these are the biological processes and the genes that are upregulated upon overexpression of CNNM1. So further, these are the specific germline specific genes that are highly enriched upon the overexpression of CNNM1. So like integrin beta, topo 2A, F2S2 are like most essential for the SSC maintenance. So further, we did an interactome by pulling out the CNNM1. So then we could found that CNNM1 was interacting with the PRL2, which is a phosphatase of regenerating liver 2. So which is found to downregulate the PTEN and has a very crucial role during the gonocyte to the spermatogonia transition stage, is being interacting with the Bateman domain of CNNM1. So to conclude my work, CNNM1 is one of aberrantly expressed gene in the male infertility, and we are the first group to report that CNNM1 expression is very strong in the brain and testes, and the primordial germ cell expression showed very less expression of CNNM1, while after the gonad formation and up to the 3 day, at the day of 3 days postnatal, it show where the high self-renewal population do exist, CNNM1 expression shooted up. So further, when the spermatogonial stem and the localization, when we did a localization, it was more localized to the SSC population. Upon the differentiation of this SSC population, we saw the decrease in the CNNM expression, while the overexpression of CNNM upregulated the germ cell population. From this, we can conclude that the CNNM1 might be having a very crucial role in maintaining the SSC population and the founder progenitors. So SSC, my take-home message will be like a SSC maintenance is a very highly complex process and coordinated with integrated cellular and molecular interactions, and there should be a delicate balance between a self-renewing population and the differentiating population to maintain a proper fertility. So CNNM proves to be one of crucial molecule that is essential in maintaining the founder population of SSC population. Okay, these are my publications and awards. I would like to thank Dr. Pradeep Kumar Ji, who is my mentor, and Dr. Malini Lavoraya for allowing me to use her lab facilities, and Dr. Harry G. Leach, who helped us in PGC isolation and culture. And these are my funding agencies. I am thankful to my lab mates.
Thank you, so good presentation and very good scientific content too. So any more questions?
Hello, Irfan. So nice work. So I just wanted to know what is the aim? I mean, in future after having this particular protein. So what is how it will help to the human civilization? Have you any idea?
See, SSC population is very essential for the formation of a functional sperm and everything. So there are the cases where we don't have like a zoospermia, where we don't have a proper sperm and all. So when as we used the letrozole, which is like now it is in a medicine, use it for a treatment of non-obstructive zoospermia and all, we could see the overexpression of CNNM. So we could speculate that it might be playing a crucial role during the process of generation of a fertile sperm.
Thank you.
Thank you.
Thank you. We move to the next presentation now. Shatrughan Upadhyay, and he's from Reproductive Endocrinology Laboratory, Department of Zoology, Institute of Science, Banaras Hindu University. You will get 8 minutes for your presentation, so you must stick to the time. Thank you.
Okay, a very good afternoon, ma'am. Hello?
Yes, we can hear.
Okay, ma'am. A very good afternoon, respected chairperson and teachers. Myself, Shatrughan Upadhyay from Banaras Hindu University, going to represent our part of research work on topic role of 14-3-3 beta protein on ovarian function, on ovarian folliculogenesis, steratogenesis, and its correlation in the pathogenesis of PCOS in mice. Like mammalian reproduction is a complex process with several critical steps like sexual maturity, reproductive cyclicity, gamete production, fertilization, and embryo development. Many factors influence this reproductive process, including hormone, cytokine, growth factor, adapter proteins, and morphogen. Among them, different isoforms of 14-3-3 proteins are believed to play a critical role in mammalian reproductive physiology and pathophysiology. 14-3-3 proteins are family of highly conserved regulatory molecules consisting of 7 isoforms in mammals. 14-3-3 proteins interact with several intracellular phosphorylated target proteins and regulate various cellular functions such as apoptosis, cell cycle, insulin signaling, and metabolic activities. 14-3-3 protein promote ovarian steratogenesis by increasing the transport of cholesterol to mitochondria. In 2012, De et al. Reported that all the isoforms of 14-3-3 protein expressed in oocyte. Further, in their investigation, in 2016, they also reported that the decreased level of 14-3-3 protein associated with activation of maturation promoting factor and release of oocyte from meiotic first arrest. Any altered expression of 14-3-3 protein associated with ovarian dysfunction like polycystic ovary syndrome. The polycystic ovary syndrome is a multifactorial endocrine metabolic disorder characterized by cystic follicles, anovulation, and hyperandrogenism. PCOS implies various severe consequences to female health, including metabolic syndrome, type 2 diabetes, and infertility. Further, PCOS is also associated with hyperinsulinemia with compensatory insulin resistance. The earlier proteomic study from our laboratory demonstrated that the level of 14-3-3 protein increases significantly during delay ovulation, and normal ovarian activity, mainly ovulation, coincided with the period, showing decreased expression of 14-3-3 protein. However, the possibilities for the involvement of 14-3-3 protein in pathogenesis of PCOS are not known. Thus, the present study was aimed to comprehensively elucidate the potential role of 14-3-3 beta protein in regulating the reproductive and metabolic features in letrozole treated PCOS mice model. In order to answer these questions, our objective was to develop PCOS mice model followed by their validation to evaluate change in expression pattern of 14-3-3 beta protein in ovary of PCOS mice, to evaluate the direct role of 14-3-3 beta protein and their regulation of folliculogenesis, steratogenesis, and their correlation in pathogenesis of PCOS. In order to accomplish our first objective, the adult female mice was taken and divided into 2 different groups. Group 1 is vehicle, while other is treated with letrozole for PCOS induction. After the last dose, mice were sacrificed and ovary and serum was collected for PCOS model validation and evaluation of expression pattern of different isoform of 14-3-3 protein. For second objective, again, adult female mice ovary were dissected out and divided into different groups. Group 1 received vehicle, while other is treated with different concentration of 14-3-3 protein. After the incubation period, culture media and ovary were collected for further analysis. In our result, we found that the mice treated with letrozole show presence of various cystic follicles in histological sections, followed by significant increase in circulating level of insulin and testosterone. In addition, the expression of insulin receptor was found to be significantly decreased in the ovarian tissue. After successful validation of polycystic ovary syndrome in PCOS mice model, we evaluated the change in expression pattern of 14-33 protein, in which we found that the circulating level as well as in ovarian tissue level, 14-33 beta protein increases significantly. In addition, to evaluate direct role of 14-33 beta protein, we perform in vitro study in which we treated adult mice ovary in vitro with different concentration of 14-33 beta protein, in which we found that the expression of PCNA, BCL2 increases significantly, while the expression of caspase 3 protein decreases significantly in dose-dependent manner. Further, the expression of star 3 beta HSD and 17 beta HSD, which are estrogenic marker, increases significantly in dose-dependent manner. In addition, the ovary treated with 14-33 beta protein in vitro condition so the significant decrease in expression of aromatase and insulin receptor. Further, the ovaries treated with in vitro with different dose of 14-33 beta protein so significant increase in expression level of testosterone and significant down level of estradiol in culture media. Thus, from above finding, it may conclude that the expression of 14-33 beta protein increases significantly in letrozole-induced PCOS mice model. The direct administration of 14-33 beta protein inhibit aromatase activity, which is associated with hyperandrogenism. In addition, the treatment of 14-33 beta also causes prolonged survival of antral follicle and suppression of ovulation. This slide represents the treatment of 14-33 beta to the in vitro condition, how causes insulin resistance, hyperandrogenism, and prolonged survival of antral follicle. These accumulatively associated with pathogenesis of PCOS in mice. These are references. Thank you.
Thank you. The session is open to questions.
What is the dose of 14-33? You have mentioned high dose and low dose. What is the dose you have given?
In low dose, we have given 10 nanogram per ml in culture media, and in high dose, 100 nanogram per ml in culture media.
Have you checked the concentration in vivo?
Sir, dose were selected according to Yubica et al. 2017 method.
Okay, so you have just reference those you have given.
Yes, sir.
So did you get bad expression also? Because so BCL2 expression is studied.
Sorry, sir.
Can you hear me?
Yes, sir.
So 14-33 beta, so heterodimer is bad protein. So
14.
The bad expression, so BCL2 antagonist conduct.
I didn't get you. BCL2 expression?
So you checked the BCL2 expression, right?
BCL2 expression increases significantly. Yes, sir.
Yeah. And another pro-apoptotic protein, so gene expression, did you study with regard to bad?
No, sir, I am not checking backs.
Okay, thank you.
Okay.
So thank you for your.
Okay, thank you, sir.
So we move on to the last session. So the next speaker is Subin Balachandran. So he is from Department of Zoology. Sacred Art College. So the title is an in silico investigation on the reproductive effects of commonly used synthetic phenolic oxidants, so butylated hydroxyenzole.
Good afternoon, everyone. Myself, Subin Balachandran. I'm a senior research fellow at Department of Zoology, Sacred Hearts College, Tevada. I hope my video and audio is audible and visible for you. My work is entitled An In Silico Investigation on the Reproductive Effects of Commonly Used Synthetic Phenolic Antioxidant Butylated Hydroxyenzole. So coming to the background of the work, that is increased infertility, as the speakers speaking earlier was telling about the cases of increased infertility cases. That is about 15% of infertility cases are reported to be of unknown factors, or no clear evidences are available about this infertility cases. So one of the reasons behind this infertility cases is supposed to be the improper feeding habits. That is, for example, in techies and all, the improper feeding habits is very high. And that might be a reason, it is suggested that it might be a reason for this infertility cases. And one of the compound we selected is BHA, that is butylated hydroxyenzole, which is a commonly used synthetic phenolic antioxidant compound. And this compound is used in food to reduce the oxidation and to increase the keeping quality of this compound, means the food items. And the ADI, that is admissible daily intake of this compound, is restricted to 0.5 milligram per day per individual. But no report still is available on a clear reproductive effects of this compound. This is the compound. It is a mixture of 2 and 3 tertiary butyl 4-hydroxyenzole, and this comes an array of compounds, means products in which BHA has been used. So here an in silico work has been designed as a screening part of my PhD work, and here mainly 3 objectives has been formalized. That is, first, to visualize whether BHA binds with selected steroid receptor. Second one was to compare the binding pattern of BHA with other standard compounds. And third one is to predict whether butylated hydroxyenzole can interfere in normal reproductive functioning of individuals. So here in methodology, we use BioVida Discovery Studio 2018, a molecular docking software platform for the work, and ligands chosen here was androgen agonist, androgen antagonist, and butylated hydroxyenzole. And the receptor used was rat androgen receptor, the PDB ID is bracketed, and the ligands used were androgen compound, that is stanosol and methylenone, and anti-androgenic compounds, that is drugs, that is oxandrolone and finasteride, and the reference molecule BHA, and the upcame ID is bracketed here. And coming to the results, the docking results are interpreted based on the mainly 4 parameters, that is lipdog score, that is a docking score, the bond length, the nature of bonding, and amino acid residues. So this is the 4, that is 2 androgenic compounds and 2 anti-androgenic compounds. The docking results has been given here. Here the nature of bonding, that is number of hydrogen bonds, number of hydrophobic interaction, this lipdog score, that is the docking score, and this amino acid residues and corresponding bond length has been. Actually, in our studies, we have chosen many compounds, that is both androgenic, anti-androgenic, estrogenic, anti-estrogenic, progesteronic, and anti-progesteronic compounds. And only a few of them, that is only 4 compounds has been shown here for reference. And the corresponding docking images of this, that is the 2-dimensional docking images of the compounds, are also shown here. So this green color, the hydrogen bond is on, and this violet color, that is hydrophobic interaction, and correspondingly. So if from this screening, we have visualized that this is the results of docking of BHA, that is our compound of interest. And this is the two-dimensional image of that. So from this analysis, we found that an anti-androgenic compound, finasteride, this compound is showing maximum similarity with the compound of our interest, that is butylated hydroxy. It is clear that the Lipdog score, first we are checking the score, that is Lipdog score of 77 and 71. They are approximately equal for the both compounds. And while checking the hydrogen bonds, the hydrogen bonding pattern in this glycine 711 is common for both. And 3 hydrophobic interactions, that is methionine 742, methionine 745, and valine 746. They are also shared by both these compounds, that is our compound of interest and finasteride. And another interesting thing is that an electrostatic interaction might be that is made by methionine 74, which is found in BHA. And a similar hydrogen bond is also found in finasteride. From this, here we can see the images clearly. That is hydrogen bond, hydrogen bonding patterns, and hydrophobic interaction. This can be similar for both the compound of interest and butylated hydroxy anisole. From these array of visualizable imaging and docking, we have concluded that butylated hydroxyanisole, it can clearly bind with the rat androgen receptors. And second thing is that it was comparable, that is the binding pattern of the selected compound was comparable with that of finasteride. And third one is that we are predicting, or this is only a predicting tool. So we predict that it might, that is the compound of interest might have an anti-androgenic effect. Coming to the discussion part, finasteride, it is an anti-androgenic compound, which is an inhibitor of 5-alpha reductase. That is, it's the 5-alpha reductase inhibitor. It reduces the dihydrotestosterone. That is, it affects the sexual dysfunctioning. That is, by reducing libido, erectile dysfunctioning, ejaculatory dysfunctioning, and so on. So we are inferring that the androgenic metabolism in a physiological system can be affected by this anti-androgenic compound finasteride. We are adding a point that molecular docking is an in silico tool and it is a screening or prediction tool only. Wet lab experiments are mandatory, that is in vitro and in vivo must be used to validate these results. And the publication, this is all the compounds which were selected by us and the results has been published. And I acknowledge Department of Computational Biology and Bioinformatics, University of Kerala for the molecular docking consultancy, and the funding was done by UGC. Thank you.
Yeah, my presentation today. So now the session is open for questions.
It's a nice presentation. Why you have checked, interaction with rat androgen receptor?
Okay, sir. Now we are conducting the animal model experiments we are doing in rat. So I have selected rat androgen receptors.
So you could have directly, seen the human, because ultimately.
So, sir, actually, I have done in human androgen receptors also. And that part is not shown here. We have done a comparative docking between both human and rat androgen receptors. And they also show similar results. But only since we are now doing the animal experimentation with rat model, we have choose this receptor. Thank you, sir.
Your Gonadal. Yes. Am I audible?
Yeah, ma'am, you are audible.
In one of your slides, you mentioned about the stenozolol. Stenozolol compound.
Little bit louder.
Stenozolol compound.
Yeah.
Yeah. What about its molecular docking? The software you have utilized?
I have told that I have used the BioVida Discovery Studio. 2018 version has been used.
Just for your information, I am telling you this recently. A Gaussian software is being utilized for this purpose for molecular docking. That is more authentic when compared to the other software packages. And one more thing, what about the reaction of the stenozol with the receptor, androgen and estrogen receptors? Did you check for that?
Yeah, ma'am. we found that they are binding, but actually in our work we are comparing with how the binding pattern of our compound of interest was with. Actually, it was in slide I showed that that is they are binding and it is showing a good binding affinity also.
Binding of similar with more with androgen or estrogen?
I have here checked out androgen. It is having high binding with androgen only.
Why?
And it is actually an androgenic compound. I hope so. Its drug is preferably an androgenic drug. That is androgenic compound.
No, for your information, I'm telling you, this is the derivative. It's a non-aromatizable androgen.
Okay, okay, ma'am.
Just check once again.
Okay, thank you, ma'am. Thank you. I'll be checking.
Yeah, so basically the interesting physical analysis you made, but BHA functionally it has its own receptor binding, and so many literatures have been published in relation with BHA. So you can't say only by docking and the posing similarity, both the compound act functionally. Okay. So
sir, actually, we are now doing cell culture and animal experimentation. As a primary investigation, only we are doing this.
Yeah. In addition, the scoring was very low. So some other the first one in the first panel, I forgot the name, but that scoring in lip score was very high. So the compound basically, if it is binding to the receptor, so the binding score should be higher. So whatever the other compounds you tested was having more than 100 kilocalories per mole, but your BHA and the other one which you are comparing and showing similarity, so you are saying the score is only 70. So
okay, sir. I hope you are misunderstand. That compounds showed high affinity was androgenic compounds, and it showed low was anti-androgenic compounds, sir.
Yeah, with regard to your scoring and then comparison. It's okay.
Yes, okay, thank you.
So if no more questions, so I thank you for your good lecture.
Thank you, sir. Thank you.
Yeah, with this today's session is completed. So again, I thank Professor Malini Madam for giving us good opportunity to share this session.
Okay, so before we conclude this session, I just have a couple of important announcements. Tonight, the transportation from here to the dinner location is going to be here at 7 o'clock. So we'll leave from here at 7 p.m. The second thing is there is going to be a bar tonight, but it's going to be cash bar, not a free bar. Okay, and the drinks are going to cost somewhere between 150 to 200 rupees. We will have a coupon counter. You can buy your coupon and then present the coupon to the bartender, upon which you can avail your drink. Okay, thank you.
Thank you, Jay, speakers and presenters for successful completion of the session. As a token of gratitude, I request Assistant Professor Ilangovan sir to present the memento, chair and co-chair. Assistant Professor Ilangovan.
ಅಲ್ಲೇ ಇನ್ನೊಂದು ಫೋಟೋ.
Thank you, sir. Thank you, Dr. Dashri Bakshi and Professor Dr. Basappa for chairing the session. Now we'll move for tea break. 10 minutes. At 4:45, the next session will start. Again, an online session, and finally, a poster presentation session will go on. Thank you.